Hey everyone, and welcome. Today, we're going to dive into a brand new AI-powered tool that's basically giving scientists a superpower, the ability to see exactly what's happening inside our cells. Let's jump right in. So, to really get what we're about to talk about, we need to start with a huge question that drives so much medical research. When a disease kicks off or a new drug starts to work, where exactly inside a single cell is the action happening? Well, it usually boils down to one of two places, right on the cell's outer edge, its membrane, or deep down in its interior. And knowing the difference is absolutely critical because a protein's location is everything. It determines whether it's doing its job or, causing a lot of trouble. Okay, but here's the catch. Actually seeing that difference, it's been a huge headache for scientists. To get why, you first have to appreciate just how crowded things get at the microscopic level. I want you to imagine looking at a big clump of soap bubbles all smushed together. It's next to impossible to tell where one bubble stops and the next one starts, right? That is exactly the problem scientists have been dealing with. Under a microscope, cells are constantly touching and overlapping, making it a real nightmare to isolate and study just one. And this is where the old way of doing things really falls short. See, older methods could only give you a whole cell view. They could tell you if a protein was in the cell, kind of like getting a text that says a package was delivered to your address. But that's not good enough, is it? For the really important research, you need to know where it is. Is that package sitting on the doorstep, the membrane? Or did someone bring it inside to the living room, the interior? That's the detail that changes everything. So, to finally answer that question, a team of researchers built a really ingenious new tool. Let me introduce you to the hero of our story, a tool called FluorSW. So what is FluorSW? Well, think of it as this super smart open source software that was built to do one thing and one thing really well. Analyze those crowded images, perfectly outline each individual cell, and then precisely measure the fluorescent signals, those are the glowing tags that let us track proteins, in two separate zones, the membrane and the interior. So how does this thing actually work its magic? It's all about the very clever toolkit it has under the hood. Let's break it down. It's basically a three-step process. First step, it uses some slick AI to find and trace the exact outline of every single cell, even in a packed crowd. Second, for each of those cells, it automatically draws two distinct zones, a skinny little ring around the edge for the membrane, and then all the space inside that ring, the interior. And finally, it gets the real numbers, accurately measuring how much of that fluorescent glow is in each of those two zones. Okay, that first step, finding the cells, that's powered by this incredibly clever AI called Stardust. It's a deep learning model that's been trained to see cells not just as blobs, but as something called star-shaped polygons. So why does that matter? Well, most cells kind of have this central body with edges that radiate out, almost like a star. By looking for that specific shape instead of just a circle, Stardust gets unbelievably good at telling cells apart, even when they're all jammed together. It's a brilliant way to solve one of the oldest problems in microscopy. But finding the cells is only half the battle. In this kind of microscopy, scientists use different colors to track different proteins, say red for one, green for another. The problem is, sometimes the signal from the green protein can kind of leak over into the red detector and the other way around. Like watercolors on wet paper, the colors can bleed together, creating signals that aren't really there. This whole mess is called spectral crosstalk, and trust me, it can totally wreck an experiment. This is where FluorSW comes in with a fantastic fix called precise mode. Instead of just accepting any color signal it sees, it uses something called dominance logic. Basically, it tells the computer, look, don't just count any faint green you see. Only count a pixel as green if the green signal is way stronger than any other color in that exact spot. This super simple rule is incredibly powerful. It stops the colors from bleeding and makes sure the data you get is clean. Okay, so once it has these super clean measurements, FluorSW calculates something that's a total game changer, a new metric called differential interior intensity. Think of it like this. Instead of trying to make sense of two separate numbers, one for the membrane, one for the interior, this metric boils it all down to a single powerful score. This one number answers a very simple question. Is the protein moving from the outside to the inside? And because it's a normalized score, things like the cell's size don't throw it off, making it the perfect way to track this critical movement. All right, this is where we get to the real wow moment. What does this new level of detail actually show us that we were missing before? Okay, so let's picture a classic experiment, right? A scientist adds a drug to a cell to see what happens to a certain protein. Now, if they were using the old school whole cell measurement, the total amount of that protein in the cell might not change one bit. Using those old methods, your results would look something like this. The before bar and the after bar are pretty much the same height. You'd look at this and go, well, I guess the treatment did nothing. But you'd be totally 100% wrong. Because when you use FluorSW's high-def, compartment-specific view, the real story just pops right out. The protein hasn't gone anywhere. It has moved. And boom, this is where you see the magic of FluorSW. Look at this data. After the treatment, the amount of protein on the membrane plummets, and at the same time, the amount in the interior skyrockets. The protein has been internalized. It's been pulled from the surface of the cell to the inside. That movement is a huge deal in biology, especially for things like our immune system. And with the old methods, it was completely invisible. You just couldn't see it.
So, okay, we can see proteins moving around. What's the big deal? Why does this new view matter so much for science and ultimately for medicine?
Oh, the impact is just massive. In immunology, this is crucial for figuring out how key immune cells get activated. For cancer research, it lets scientists get a much better handle on immune checkpoint receptors, the main targets for some of the most exciting cancer drugs out there. In drug development, it's a clear way to see if your drug is actually doing what you designed it to do. And maybe most importantly, because it's an open source standard tool, it just makes the science better and more reliable for everyone. I mean, the researchers who built it said it best. They explain that FluorSW finally addresses the common pain points in this field. It lets them tell the difference between a real change in where a protein is versus other confusing things like the cell just making more of the protein or getting bigger. It just cuts through all that noise and gets right to the point.
What we have here is a brand new window into the secret life of our cells, something we've just never had before. It really makes you stop and think. Now that we can finally see this invisible dance of molecules moving from the doorstep into the living room with this kind of clarity, what huge medical mysteries are we going to solve next?